Well, hello there and welcome back to our series of videos on the topic of cell division. Now, we all know that sexual reproduction produces genetic variation. Here we have our father and mother and their whole family of children. And you notice that all their children look different, right? There's variation. And variation is important because it increases the chance of a species surviving in a changing environment. If all my offspring are genetically identical, then a change in environment might cause all of them to die because they're all equally bad with coping with that new environment. But if I have genetic variation, then if the environment changes suddenly, well, some of my offspring may still survive, while others may die, and therefore my species is able to survive overall better. But the question is, how does sexual reproduction produce genetic variation? How does it do that? Now, the secret lies actually in meiosis. Meiosis is an important part of sexual reproduction because meiosis produces gametes, right? And gametes are needed for sexual reproduction. So something about meiosis helps to contribute to genetic variation. And that's what this entire video is going to be about. In this video, we're going to ask ourselves the question, how does meiosis lead to variation? Now, of all the slides in this video, this is the single most important slide. So if you don't understand anything else, just come back to this slide and understand the points on it. So what helps to produce genetic variation in an organism which reproduces sexually? Well, the first thing is meiosis. Meiosis helps to produce genetic variation. And that's because meiosis produces gametes which are different. Gametes which are different have different combinations of genes and alleles. So imagine the sperm or the eggs in a person. And imagine the sperm in me, okay, for example. Imagine if all my sperm, all my gametes were genetically identical. Well, if all my gametes were genetically identical, then all my offspring would be identical. I wouldn't have any variation. But that's not the case because all of my sperm, all of my gametes are different. They all contain different combinations of genes and different combinations of alleles. And that's why my offspring are different. And how does meiosis produce gametes that are different? It does so through two different ways. The first way is crossing over. And the second way is the independent assortment or independent segregation of homologous chromosomes. We'll take a look at that in the next few slides. Okay, so the first important point is meiosis helps to produce genetic variation because meiosis produces gametes which are different, which are genetically non-identical, and it does so through two processes, crossing over and independent assortment. And secondly, genetic variation is also produced through random fertilization. So once I get different gametes, so let's say, there are many different gametes inside me, all my sperm are different, and then there's many different gametes in my wife, all her eggs are different, right? I randomly pick one of my sperm and I randomly pick one of her eggs, and that's random fertilization. So that's another reason why all our offspring are different. It's due to the randomness of fertilization, right? Or randomness of which sperm fuses with which egg. Okay, so 3 points to remember. What helps to produce genetic variation? First, crossing over in meiosis. Second, independent assortment. And third, random fertilization. So I'm going to go deeper into each one of these in the next slide. Okay, we're going to start with independent assortment. So I want you to consider this simplified example again, right? So this is a simplified cell. Imagine that this is Mr. Lim. I have 4 chromosomes in my cell. Okay, I know normally I have 46, but imagine I have 4. Okay, I have 4 chromosomes and they are paired up into 2 pairs of homologous chromosomes. So the yellow one represents those from my father and the white one represents those from my mother. So my father gave me this combination of alleles. He gave me a capital E, capital A, and capital B allele, right? So it could be like dark hair, dark eyes, black hair, black eyes, and something else. But my mother gave me this combination of alleles. She gave me the small e, small a, and small b allele. Now imagine that you zoom in on my testes. It's a bit disgusting, but imagine you zoom in on my testes and you look inside and you see how the sperm are being produced. So there are some cells which are dividing by meiosis to produce sperm. And some of these cells will be in metaphase 1. And remember what happens in metaphase 1. The pairs of homologous chromosomes line up along the equator. Now I want you to imagine that the pairs of homologous chromosomes line up this way. That all of my father chromosomes line up on one side and all of my mother chromosomes line up on the other side. What kind of gametes, what kind of sperm will we produce? Well, if all my father chromosomes line up on one side, then I will produce sperm with combination of capital E, capital A, and capital B alleles. And I'll also produce sperm with a combination of small e, small a, and small b alleles. Right? So this is the first combination. But what you realize is that this combination is exactly the same as what my parents gave me originally. So there's nothing new, right? My father gave me big E, big A, and big B together. That's the sperm that he gave me. And now I'm also producing a sperm with the same combination. My mother gave me an egg with small e, small a, and small b. And now I'm producing a sperm with the same combination of alleles. So there's no new combinations. However, that's why independent assortment or independent segregation of homologous chromosomes is so important. Because it doesn't have to be that all my father chromosomes line up on one side and all my mother chromosomes line up on the other side. Because each pair of homologous chromosomes segregates or assorts independently. So this pair can decide that father's on this side, mother's on this side, but this pair can decide that father's on this side and mother's on this side. So with this, I can end up with a different combination of alleles. Because now, on this side, I will end up producing sperm with big E, big A, and small b together. And I also end up producing sperm with small e, small a, and big B together. And this combination of alleles is different from what my father and mother gave me. So this is variation. There's now a new type of gamete being produced, right? So I'm producing different gametes. I'm producing some gametes with this combination, and I'm producing some gametes with combinations similar to my parents. So it is this new combination of alleles that results in genetic variation. Because now I have produced many different types of sperm. Some of my sperm have the same combination of alleles as my parents, but some of my sperm have different combinations of alleles from my parents. So there's new variation due to independent assortment of homologous chromosomes during metaphase 1. But at this point, if you're thinking very hard, you realize that actually there's certain combinations that have not been made yet. And that's because certain alleles are found in the same chromosome. So if you take a look here, big E and big A will always end up on the same side, right? No matter whether I put the yellow one here or the yellow one here, big E and big A will always end up on the same side. So how can we separate big E and big A and make them end up in different gametes? And that's where crossing over comes in. Remember that crossing over takes place during prophase 1, where pairs of homologous chromosomes pair up closely through a process called synapsis, and then they can cross over at points called chiasmata, and they may exchange lengths and parts of DNA. So they may exchange lengths of DNA with one another. They may exchange alleles with one another. So you can see over here, and that produces a new combination of alleles that was not found in my parents. Because my father gave me big E and big A together. But now, because of crossing over, some of my sperm will contain big E and small a together. right? So that's a new combination that was not found in my parents. So again, this is another source of genetic variation. So we have seen that independent assortment and crossing over helps to produce many different types of gametes with different combinations of alleles, and that contributes to genetic variation. Now let me blow your mind with how many different possible combinations or types of gametes can actually be produced. You see, if you just consider independent assortment alone, if let's say I have two pairs of homologous chromosomes, okay, so one pair here, right? Actually, there's already 2 to the power of 2 possible gametes formed, right? So if you take a look here, this is possibility 1, where red, blue goes to one side and blue, red goes to the other side. We get this. So we get this kind of gamete here. Possibility 1, possibility 2, right? Blue, red, red, blue. But we get this independent assortment arrangement at metaphase where all the reds are on one side and all the blues on the other side. Then you get combination 3, where it's red, red, and combination 4. So if you have 2 pairs of homologous chromosomes, we have 4 possible gametes, 2 square, right? So if we have 23 pairs of homologous chromosomes, like in human beings, there are actually 2 to the power of 23 possible gametes, right? Which is 8 million possible gametes can be produced. And that's just with independent assortment. I haven't even considered crossing over yet. So that brings us finally to random fertilization, right? Imagine, I can already produce 8 million possible sperm, right? Due to independent assortment. And my wife can produce 8 million possible eggs. Now, during fertilization, only one of these eggs will be fertilized by one of these sperm. So random fertilization contributes even more to genetic variation because, I just, of all these 8 million possible eggs, I pick one. Of all these 8 million possible sperm, I pick one and they fuse together. So there's like 8 million times 8 million possible offspring produced. And that's a whole lot of genetic variation. So let's sum up again. The big question is, what helps to produce genetic variation? Or how does meiosis contribute to genetic variation? And we've seen that meiosis helps to contribute to genetic variation because it produces genetically different gametes with different combinations of alleles. And it does that through, firstly, crossing over, which happens in prophase 1, and secondly, independent assortment, which happens in metaphase 1. And on top of that, we also have random fertilization, which further helps to increase the amount of genetic variation. Now, before we end, just one thing for you guys to do on your own over the break, which is to compare mitosis and meiosis, and to complete the comparison table in your notes. So you can look at textbook page 312 to help you, and we'll discuss more of this in class.